Hello students, today again we are going to continue with biology chapter number 4, molecular basis of inheritance. In this topic, last lecture we have covered the topic central dogma of protein synthesis. Then we have studied about reverse transcription. Then we have studied the topic of introduction of transcription. Today we are going to study about the topic transcription in detail. Okay, we will start with the definition part. Okay, transcription is the synthesis of mRNA from DNA which occurs in 5' to 3' direction. Okay, when I say transcription, it means in simple words we can say that transcription means the genetic information is copied from DNA to RNA. Okay, genetic information is copied from DNA to RNA. This genetic information is in the form of coding sequence, codons. Okay, during transcription, a DNA sequence is read by a RNA polymerase. This RNA polymerase is an enzyme which catalyzes this whole reaction. That means the genetic information, the genetic transformation is carried out with the help of an enzyme, that is RNA polymerase. Okay, RNA polymerase brings about this reaction, and the new strand that will be formed, mRNA strand, is called as complementary strand, and it is anti-parallel to the coding strand. Okay, transcription is the first step that leads to gene expression. Next, when I say transcription, it involves transcription unit. In short, I have told you about the transcription unit in our last lecture, but today I will be explaining this in detail. Transcription unit consists of promoter, promoter, structural gene, terminator. When I say promoter, that means promoter is located at 5' end. That is upstream. Okay, promoter is located at 5' end, that is upstream. Okay, it provides, what is the function of promoter? It provides binding site to the enzyme RNA polymerase. Promoter provides the binding site to the enzyme RNA polymerase. Okay, and it is located at 5' region. Next, when I say structural gene, what do you mean by structural gene? The two strands of the DNA are having opposite polarity, shows opposite polarity. Polarity means direction. Okay, the two strands, the strand that see here in this diagram, the strand that runs in 3' to 5' direction, and the other strand runs in 5' to 3' direction. That means both the strands shows opposite polarity, opposite direction. When I say the strand that runs in 3' to 5' direction, it is called as template strand. The other name for template strand is antisense strand, and this template strand participates in the process of transcription. That means it is involved in the process of transcription. And the other strand, that is the strand that runs in 5' to 3' direction, is called as coding strand, and the other name is sense strand. This strand does not participate in the process of transcription. Okay, and RNA polymerase catalyzes this whole process of transcription in 5' to 3' direction. Next is terminator. When I say terminator, what does this mean? Terminator, it is located at 3' end. Okay, terminator is located at 3' end, that is called as downstream of the coding strand, and terminator terminates the process. Okay, it terminates the transcription process. Next, when I say transcription unit, we need to study two more here concepts, that is polycistronic and monocystronic. What do you mean by polycistronic and monocystronic? Cistron means it is an old name for a gene. Okay, cistron means an old name for a gene. Polycistronic means they are found in prokaryotic genes and monocistronics are found in eukaryotic gene. When I say mono means single. One promoter directs the synthesis of one mRNA and translates to only one protein. Okay, one protein is formed. Mono means one protein. And polycistronic means here also one promoter will direct the synthesis of one mRNA, but that will be translated to more than one gene, one polypeptide. That is polycistronic. Okay, mono means one protein will be transcribed and poly means more than one gene, that is polypeptide will be transcribed. Okay, we need to study about the split gene. Okay, the structural gene are composed of two things, that is coding sequence and non-coding sequence. When I say coding sequence, you can see here, okay, coding sequence and non-coding sequence, okay, from the numbers that is written 1, 2, 3, 4, 5, 6 and 7. These all are coding sequence, coding regions. They are also called as exons. And the in-between region that are present, that are labeled with the alphabet A, B, C, D, E, F, G, they are called as non-coding sequence and the other name is introns. They both are alternatively separated from each other. Okay, coding sequence are the sequence that appear in the processed mRNA, whereas non-coding sequence does not appear. Okay, they are expressed, they cannot be expressed. Next, we will study about the steps of transcription. When I say transcription, we have studied transcription unit, and the next is steps of transcription. It consists of 3 steps: initiation, elongation, termination. Okay, it consists of 3 steps, that is initiation, elongation and termination. What do you mean by initiation? Let us study. I have told you RNA polymerase binds to the specific DNA region and then the process of initiation, transcription starts. This is called as initiation. Okay, see here in this diagram you can see RNA polymerase binds to the 5'N, that is promoter. Okay, 5'N, that is called as promoter, and it brings about the process of initiation. After the initiation process, these 2 strands are separated from each other. You can see here this sigma factor is also there. Sigma factor helps to identify the enzyme RNA polymerase and attach to the promoter. Okay, after the step of initiation, elongation will take place. What do you mean by elongation? Elongation means according to the base sequence, after the separation, after initiation, the 2 strands are separated from each other. Okay, after the separation, what will happen? According to the base sequence that is present on the template strand. Okay, I have discussed about the template strand and coding strand. So as per the base sequence that are present on the template strand, the complementary RNA nucleotides are selected and joined one after the other and forms mRNA. Okay, and forms mRNA, you can see here. Okay, then the last step is termination. When the RNA polymerase enzyme reaches the terminator end, okay, it reaches the terminator signal, both the enzyme, that is RNA polymerase, and newly constructed RNA, both is released, both fall off. We can say that both are falling down. Okay, when it reaches to the terminator, the remaining RNA polymerase enzyme and newly constructed RNA both fall off. Okay, and here in this termination process, rho factor is helping. In initiation process, sigma factor is helping, whereas in termination process, rho factor help out. Okay, now when I have discussed about the polymerase here, RNA polymerase enzyme, we need to discuss it in both prokaryotic organism as well as eukaryotic organism. Okay, now we are going to study about RNA polymerase. Okay, RNA polymerase, when I say in prokaryotic organism, only single RNA polymerase is present. That is RNA polymerase. Okay, it is having only one type of RNA polymerase, and it does not require the prokaryotic organism. The mRNA that is formed does not require any further processing. Whereas in eukaryotic organism, three kinds of RNA polymerase are found. What are those three kinds? Let us see. RNA polymerase 1. Okay, it is located in nucleus. RNA polymerase 1 is located in nucleus and synthesizes the rRNAs. It synthesizes rRNAs. Then RNA polymerase 2 is located in nucleoplasm and it synthesizes hnRNA. That means heterogeneously nuclear. Okay, what do you mean by HN? Heterogeneous nuclear RNA and mRNA. This RNA polymerase 2 is used in the process of transcription that we are studying right now. And RNA polymerase 3 is located in nucleoplasm and it synthesizes tRNA as well as small nuclear RNA. Okay, small nuclear RNA. Now, when I say prokaryotic and eukaryotic organisms, see here this diagram. Okay, prokaryotic organisms do not require any further processing. Okay, the mRNA that is formed in prokaryotic organism can be used as it is. It is functional, but the RNA processing only occurs in eukaryotes. Okay, we need to process the mRNA because it is non-functional RNA that is formed in eukaryotic organism. So we need to do the process of processing. Okay, this mRNA processing involves three steps, that is capping, splicing and tailing. Okay, capping, splicing and tailing. What do you mean by splicing, capping, tailing? Splicing means removal of introns. You can see here in this diagram. Okay, first step is capping. Capping means addition of methyl guanosine triphosphate. Addition of methyl guanosine triphosphate, 3-phosphate group. So triphosphate, methyl guanosine triphosphate at 5' end is called as capping. Then splicing means removal of introns. As we have already discussed this, what are exons and what are introns? Exons are those genes who can express themselves, whereas introns are those genes who cannot express themselves. So we will remove all the introns from here and all the exons will be binding with the help of the enzyme DNA ligase. So in splicing we will remove the introns. Removal of introns is called as splicing. And the last step is tailing. That means here we are going to do polyadenylation. Okay, that means addition of adenylate residue at 3' end. What is tailing? Addition of adenylate residue at 3' end. It is also called as polyadenylation. And finally we are going to get a processed mRNA. Okay, that is messenger RNA, processed mRNA. This processed mRNA then is transported out of the nucleus through nuclear pore for the process of translation. Okay, this processed mRNA is then, after the processing, that is splicing, capping, tailing, after it is getting processed, it is transported out of the nucleus through nuclear pore for further process that is known as translation. Okay, so but this is not required if it is a prokaryotic organism. In prokaryotic organism, the steps of initiation, elongation and termination, that's all. That is going to give us the functional mRNA. But in eukaryotic organism, we are going to get non-functional mRNA. So to make it functional, we are doing the three steps, that is splicing, capping, tailing. And then the processed RNA we are getting, that is transported for the process of translation. Okay. Thank you.